Welcome, everyone. Really excited to talk today about CFT. We've worked on this for a long time, and many people on the call are in the acknowledgements. It's a long list of people that have put effort here. So I'll be talking about block phase tomography or CFT specifically in drug discovery and development application. And I really think of this in some way. When we started doing rodent nuclear imaging and PET and SPECT, we kind of would call it ADME DMPK 2.0, and I think of this as ADME DMPK 3.0. So from an outline perspective, I'll talk about kind of the whole scale, because I think scale and is really important to CFT. It really bridges a lot of gaps in the middle of pathology and radiology and opens up access to tissue research that other techniques do not. So then we'll talk about the Xerra system itself, a bit of history of how we got there, and some applications. Then we'll do a deep dive on comparing CFT with other molecular imaging techniques, primarily nuclear. And then we give a deep dive on an example. So this is really, I have shown plots like this a lot, and I think as have others, looking at the resolution spectrum versus mass for imaging, for molecular imaging. In this case, to the upper right, we think of clinical imaging as a milliliter sport. We think of the microspect and micro CT space as a microliter sport. We see a CFT image there of a brain that we're going to talk about later. That's a nanoliter sport. And we think of digital pathology as a really femtoliter sport. So we have a huge spectrum of range here in what we can study. And I show a cool image here, a quantitative immunohistochemistry image that I love from Koenig Minolta's QuantiCell that I tee into the next slide, which is if we keep going down the scale to the attoliter and we look at electron microscopy, you can actually see the fluorescent nanoparticles that Koenig makes on the tissue. That's how they make it quantitative. They calibrate in the electron microscopy range and then back have a lookup table to get back to be quantitative. And then all the way down, we have a very cool video that doesn't play here, but I recommend. I don't know how they did this, but the University of Cambridge made a video of the chromosomal structure in a mouse real-time movie of gene expression, which is at the octoliter range. So we have, as a research community and as a clinical community, really broad ranges of what we study. From a biological interrogation range, I just showed that 10 to the 26th dynamic range from a whole body down to a single variant. I think there's 37 trillion cells in the human, so it's important to have a pretty good range. In terms of disease area, we cover it all. In terms of workspace as researchers, from discovery through ultimately being in clinical practice at a hospital. Chemistry is paramount to everything we do in molecular imaging and beyond, from whether it's small molecular medicinal chemistry to GMP, CMC. And then I think as a species, we've really filled out the entire electromagnetic spectrum. It's actually interesting to think there's kind of no aspect of the electromagnetic spectrum that we don't use at some point for some assay. And today, I'll really be focusing on cryofluorescent tomography, which is incredibly useful in kind of tissue, cell, organelle, protein research. Useful, I think it's a discovery. We're going to be talking about rodent discovery research today. Useful across all disease areas. We have done some first-in-human work, and that could become more common. But primarily, this is in laboratory research. And all aspects of chemistry will be covered today. So much work done in protein engineering, cell engineering. You're as good as your chemistry. And we'll be focusing on CFT, but we'll compare the modality to others. So just by a little bit of history of the technology, many groups have worked on block phase tomography. We started working on it at in vitro almost 8 years ago. And you can see on the far left, that's a full cynomolgus head. And at the time, to be able to make a 3-dimensional RGB image like that was it is still computationally intensive, but it was really just kind of for the first time you could make an image like that. That was 40 micron sections on a cryomacrotome from Leica, imaged at each section 1800 by 1800 by 1800 isotropic voxels in the RGB. In that study, we also had a radionuclide on board, and we stopped every 10 sections and did autoradiography and built a 3D image. I don't show that image here, but it was a very telling study. And this was a kind of a breakthrough to be able to see this anatomy, in this case, pretty painstaking and tedious work, but very important and really helpful. Soon thereafter, we started putting a fluorescent imaging system on top of sectioners, multiple different types of sectioners, macrotomes, cryostats, and imaging as we section through the block. And you see some of the early images here that were right away, we knew we had something incredibly useful for studying biodistribution and pharmacokinetics, pharmacodynamics, and all sorts of modalities. And I'm going to kind of step you through how the modality works. So the system itself has come a long way. The team has done a phenomenal job. Many people, dozens of teammates, more, maybe 40 people have touched the system in some capacity, working to build it out. There's 10 of them now installed at labs around the world, and really just kind of hitting its stride in terms of adoption. It's a quantitative modality. We'll talk about that. It's a super cellular, but really high resolution imaging system. We typically section around 40 microns, but it can go down to 10 microns. It's got a 12 megapixel camera. It can really image any fluorophore. So there's 6 lasers and 7 filters. Not much you can't image off the block. So I'm going to step you through what does a study look like. You typically, there's a system, there's a picture of the most recent installed system. You can, there's different block sizes, so you can block across a range given whatever study you're running. You can put as much as you can in the block. We'll talk about that later. And your block size will change the resolution, the reconstructed resolution of the system. There's a look inside the system, so you can see the blade system and the block, and it slides along. There's the imaging tower on top is where the optics are, and you section and take an image at each depth. You do have contribution of the fluorescence from below the surface, so you will get excitation below the surface, and you do then have to make a reconstruction. The resolution and the segment, it's still mind-blowing to me, in terms of the sectioning 10 microns, and then you think about a microtome going to a micron. This is high precision performance. And in this case, you don't need to decalcify. A cryomacrotome does full frozen rodents. You image in the RGB, and then you can image at any wavelength you want. And so you get, depending on how you've configured your study and what fluorophores you're trying to image, you'll have multiple images that you can reconstruct and co-register at the end of the study. To put things in context there's going to be a lot of these plots today. So I'm not going to apologize, but there's a lot of high level modality plots. And to put things in perspective, in optical imaging, we will show a few plots from the review article we wrote with Richard Hargreaves on imaging. And you see, this was before CFT when we wrote this. You can see in the bottom left, that's the resolution versus the mass of tissue that you're interrogating. And then in the upper right, you're seeing all the in vivo fluorescence technologies, including fluorescent guided surgery and just regular laboratory surface fluorescence imagers. And there's a gap And what's exciting about CFT is you have a broad range of resolution and mass that one can interrogate. So it really bridges the gap between imaging, in vivo optical imaging. The same holds true for microspect, micropet, and microscopy. And there you see the bottom line is when you're imaging at in-plane resolutions of tens of microns and sectioning at tens of microns, you really get an incredible image of the biodistribution of the distribution of the fluorophore in the animal, as well as the really actually, I think we're getting more and more data out of the bright field images, but the RGB images also are very striking. There's no, we all love anatomical imaging combined with functional or molecular imaging in PET, SPECT, CT, but there's no better anatomical image than a 3D RGB image of the subject matter. In terms of comparing this, it's not a fair comparison to look at surface in vivo optical imaging, though it is a standard practice. There you see a bioluminescence image, and that same cell line had been transfected with DS-red, and you're now looking at the distribution of the cells in 3D from the CFT. So to be clear, the animal on the left is very alive, and the animal on the right is very dead. But it really has opened up eyes. That's pretty cheesy. It has really kind of shown us a lot of things that we had not seen in these subjects. Very often you would resect, or you might take a section, put something on a microscope, but you're really missing the whole body distribution of what you were trying to study. And I think therein lies the value of the technology. I showed you bioluminescence, and in a comparison here, we're looking at fluorescence. So in this case, the cell line had been transfected with TD tomato, and the protein, a HER2 targeting protein in this tumor model, had been labeled with an AF647 fluorophore. And there's your surface image. You definitely know there's tumor activity and probe activity and compound activity in the center of the mouse, but it's just, again, wildly informative to look at what's really happened and co-localize the cells with the imaging agents. I'll talk more in the major example of kind of downstream processing. I just wanted to kind of show off literally sort of the highest level power of doing this technique. There's been a lot of work done in what we often call pharmaco-expression. So trying to use, in this case, an AAV9 that has been transfected with GFP. And we're sectioning through, you see the bright field images. On the left, you have a control animal. There's green in the gut. There's porphyrins there. And on the right, I think it was 4 weeks later, I believe Hemi did this, and I saw his name on the call, so he can hold me to anything. But you're seeing where was the transduction? Where are we seeing now where was gene expression modified? And it's a striking image. You can see obviously a lot of hepatic, a lot took place in the liver. You can see the dorsal root ganglia. You can see the sciatica. And no one had really ever seen these things before. It was really an exciting breakthrough. And a lot of work has been done with gene therapies on the system to date. So I'm excited, and I was kind of thrown off there by my microphone problem. But I just want to add again, in terms of that history, I want to thank so much of the work that was done in vitro prior to the building out of EMIT, and I see there's a lot of in vitro folk on the line. So it's been actually a long time that we've worked on this modality and improved it. And there certainly I don't know the count now, but 50, maybe 100, 150 studies using the technique. So while it's new to roll out into general use, it has been worked on for quite some time. And here you see two tracerous images injected simultaneously, and you see different stages of renal clearance. And then from the anatomy, you're able to section out the vasculature of the kidney. So can be the number of fluorophores you can image, and the ease of doing that is very, very nice. There's a site at UCSD, Eric Ahrens. He has a very nice platform of perfluorocarbon nanoemulsions, a nanoparticle. You're looking at tumor-associated macrophage in a model with a GFP-expressing cell line, and they have this V-sense red on his compound. And you can. I mean, it's striking. You're looking at the lymph system. It's just a lot of information. I would actually even say that the MIP, I'm not sure how it's showing up on your screen. In practice, the MIP, the spinning rendering on the right of the co-registered tumors is even nicer if you fly through the data set. So very high resolution. And we'll talk about how you kind of bridge the gap to microscopy in a moment. In terms of throughput, there's a lot of ways to. It is a not a quick sectioning process. It takes many hours to section the block, 4 to 8 hours or longer, depending on if you set it at the highest resolution. The flip side on resolution is you can put a lot of animals in the block. I expect people to be putting a lot of organs in a block. This is not fluorescent work. This is very, I saw Jenna Sullivan on this. Jacob has a lot of people put a lot of effort into this protocol, which was in this case C14 imaging in a block of 80 mouse brains and then co-registering them. So that's a 3D C14 image. As this is a very powerful technique, but incredibly hard. I don't want to say tedious, but it's hard. This is not something it's not a casual sport. You wouldn't just do this every third day. But for the fluorophores, it's much easier. You can, anything you want to image fluorescence, you can block it and reconstruct it. And I foresee in the future the breakout and comparators of organs using this technology, or tissue resections, just blocking more tissue, setting it and letting it run, and then producing data. I think that's going to be a standard protocol, even if it's just resected tumors, or it's just, you can get a lot of data. You can get a whole cohort, you can get a whole study in one block. And I think that, as you see here, this is a very well powered study, actually. And I think that's really speaks to the future of the modality. And very nice data processing there. That should give the IA team from Invicro a shout out. You're looking at voxel-wise comparators across all cohorts in a common co-registered space. So I'm now actually going to give the kind of a deep dive application of what I see the most. One of my favorite examples. It's from a few years ago, but I think it is kind of the ultimate PK/PD strategy for studying a drug. And I'll go through why. This was published by Kurt Mazer. This is work that was collaboration between Ionis, Biogen, and Invicro. And the goal was to study the pharmacokinetics and pharmacodynamics of a antisense oligonucleotide. And this really is multi-resolution and multi-temporal. Here you look, the first thing done, there were two ASOs, a control ASO and a GABA1, or something that's going to would knock down GABA expression in the brain. And the different cohorts involved an intrathecal injection. The team had both capacity to iodinate with a prosthetic group, the oligo, as well as had already done a lot of work with the fluorescent oligos. So we were studying both radioactivity and fluorophores here. What's very nice in this image is, you really get the temporal organ level understanding of what's happening. What's the general PK? What's the general biodistribution of my compound? And at the 7th day, you see a autorated luminogram that actually has resolution pretty analogous to CFT, but it's one slice. And so this is a microliter sport. And the next activity, and there you see we can't, I think someone, I've been accused of showing too many spinning mice, and that's totally fine. I still like them. And now, in this case, we resected brains and sectioned them and imaged them, taking down rats at different time points. There you see the fluorescent labeled oligo as a function of time in 4 different rats and a cross section. We get the whole brain. In this case, we have really a relative quantification of concentration of fluorophore per region of brain, applying a brain atlas to the tomograms, the 3D CFT images, and then looking as a function of brain region, what was the biodistribution over the course of the study. I think it was very informative as to, it's pretty clear where it's not going in the striatum there, but it's really informative as to what was the penetration and at what concentrations. And now I think what is exceptional about understanding the full 3D biodistribution of the fluorophore in the test subject is to then shift to fluorescent microscopy. The images on the right, in that case, the tissue was transferred directly from the CFT experiment and imaged under a fluorescent microscope. So you can see the ASO plus some counterstains. The image on the left is now looking at a separate cohort, but with the same dosing, where the animals were not there was no CFT performed, but we could section, we knew where to look, we knew what to look for, and could put it under the microscope. Hemi and Jenna are on the call. I know that they put a lot of people put a lot of effort into this, and I think it's a phenomenal example of biodistribution research at multiple levels. You're covering 5, from cellular to whole animal biodistribution. In this case, there was also a great PD readout. So given that the ASO was targeting to knock down GABA expression, we were able to image these rats at 4 weeks, or actually longitudinally, with flumazenol, and it's, I think, striking. You can see visually on the left the knockdown and the drop in uptake of the flumazenol, and then the image on the right is voxel-wise area under the curve of the flumazenol, and the bottom plot is the relative comparison between the two cohorts. So very statistically significant and very well done to see not only where did it go, but what was in this case the drug effect. I'm going to now move on to a conclusion page. It's a great tool. I wrote that. That's really insightful. So now I'm going to get a little more technical and frame CFT as to where it fits in the context of rodent imaging compared to other modalities. Out the gate, I want you to know I'm not a believer in one modality trumps all. That's often a hype that happens. It's just not the reality. I think, as we stated at the beginning, you need to understand all the modalities that one can have available to them, and choose the right tool for the right task. But I will say that CFT, as you're going to see, really checks a lot of boxes in terms of value. These are two not that old, but review articles we wrote about on imaging. The first on the left was really imaging antibodies in mice, and we did a lot of work on the tables of the properties of the isotopes in SPECT and PET. I just showed the SPECT component here. And when you're choosing a SPECT and PET isotope, you have to factor in half-life. What's the energy of the gamma that changes the imaging properties? What's the annihilation distance of the positron? What are the how many gammas and positrons are emitted per decay? What's the specific activity and of the radioactivity? what's the setup of the actual system that you're doing? What's the system sensitivity? They're configurable and they vary. And if we think about counts per second per microcurie per cc, you really have to take that into account. And oh, by the way, we're doing biological research, so all things are not equal. I put this here just to say I think it's not a simple conversation to understand isotope selection and the impact on your image quality, and it really varies on the task of what you're doing. What is clear is on the right is as much, the microspect and the micro tomography instrumentation was 20 years behind the clinical because largely due in part that rodents are a couple thousand times smaller than humans. And as strong as the relative resolution was on the first slide, you really have a mass issue in rodent nuclear imaging. You're never going to be able to have relative masses, mass per subject, massive compound per subject mass. As you go down in rodent in species size, you're going to have to give more radioactivity to achieve the resolutions you need for the system to work, and it is significant. The green there is very relevant. That's quantitative whole body autoradiography, which really inspired the development of the CFT system. And then to the far left, you have an analog of fluorescent microscopy and micro autoradiography. And in those cases, you really have tremendous sensitivity from a mass perspective. Again, it's an ex vivo sport, but you can really achieve parity in terms of mass dose. And that's why historically they've been used so often for tox studies for comparator to with C14 and tritium for comparator to clinical translation. But they are also very hard. They're not for the weak of heart. Microautoradiography is you have to be trained in the art. It's a skilled, and QWA is a significant infrastructure and skill set to develop. When it comes to fluorophore selection, I won't. It's still hard, and there's a huge range in fluorophores, but and you have to give thought as to your strategy, and especially it's a lot. And you can do multi-isotope imaging, but multi-fluorophore imaging is wildly easier. It comes down to the brightness is proportional to the extinction coefficient times the quantum yield, and typically as you trend to the NIR, your extension coefficient goes up and your quantum yield goes down, and you net out at some comparator, but you absolutely have to take this into account as to what is the brightness. You do have problems. I mean, radioactivity, working with radioactivity is both great, but very challenging. There are challenges. You can quench, you can photobleach, but by and large, it is a lot easier to work with fluorophores, and they are often incredibly well characterized. So now really getting into what are the mass comparisons between fluorescence and nuclear, and specifically CFT. And this is really just a guiding principle plot on the left. And it's funny, we used to talk a lot about how when you go down, and I'm going to explain the supporting plots, but if I look at the yellow curve, which is the radioactive signal curve, and the blue curve, which is the fluorescent signal, you could argue their relative height. So that's the point. It's just a general takeaway of when you're transitioning to high mass, you will start to compete off with cold at the target in the case of radioactivity. And we see that in this plot from Tolmachev, where we looked at anti-HER2 antibodies. We had a high expressing cell line and a high Bmax and a low Bmax. And as we increase the mass of injectate, you're going to compete off faster in the low expressor. And so you absolutely have to take this into account. The problem is you still have a map, you can still have you just don't have that problem. And in fluorescence, it's just much easier. You have a fluorophore. When we're radio labeling compounds, we have one radionuclide per 1,000 to 5,000 to 10,000 molecule of compound that we're working on. When we're doing fluorescence, we almost always have a 1-to-1. We not only have a 1-to-1 labeling, but each fluorophores can provide a lot of photons for us in our collection. So on the right there, you're seeing in the 30 micrograms in this Thurber and Weisslater paper, you're at the subsaturating condition, and as you ramp up to 300, you just increase the signal. You will saturate it eventually, but you're not going to have a problem with the fluorophores that are present you're not going to have your colds competing off the hot. I would say, personally, when I used to think about this program, I really thought, well, as you trend down, you do well with nuclear always, and you will start outperforming from a signal perspective the optical. But if you go all the way down to the point where if you're trying to put 150 to a few hundred nanograms of small molecule into a mouse, you just can't get the radiolabeled specific activity to work. Thus, you go to a cut and count experiment where as I showed with ex vivo, when you put in a well counter or gamma counter, your sensitivity goes way up, but you've lost the ability to image. With the CFT system, you can image in the tens of nanogram range and for a whole mouse. And we're working. We're excited. We started characterizing this. We're confident at those mass levels. We'll see how low we go. And of course, there's lots of caveats around distribution and what your task is and so on. But the bottom line is, you're going to run out of room on the nuclear side before you run out of room on the fluorescent side. That's very exciting. Taking kind of a macro perspective of sensitivity versus interrogation scale, and I'm sorry, I hope you can see this well on your screen. It's a little blurry. This was, again, from that review article where we tried to map out sensitivity according to these different modalities. And, PET and SPECT have great translation. We'll talk about that. That's very clear. Each modality has its regime where it works, its pros and its cons. I will just say, I put in CFT here. We have a huge range of a huge interrogation scale, and we have incredible sensitivity and incredible dynamic range. And we'll talk about the dynamic range right now. In the case of when we think of DICOM data, it's a 12-bit data set. We're mapping here. We're collecting on a 12-bit. We take many exposures of the camera on a 12-bit camera and then mapping to a 32-bit system. So just first off, from a computational perspective, we have a larger dynamic range by definition. We also have a larger dynamic range because I think we all know and have all experienced, if we have a very hot region of interest in any nuclear tomography modality, the bladder will blow out anything in plane in the imaging system. You're just going to have the kid. It's very hard from the dynamic range of a nuclear system, not just because of the 12-bit, really just because of the way we're collecting the impinging photons in the detectors themselves and reconstructing them. You're going to have artifacts associated with hot and cold, and that limits the dynamic range of a PET, micro PET, or microtech, or clinical PET, or it's a general tomographic issue. That you can have that problem in CFT. There is a limit to the dynamic range, but by and large, and I think a lot of the work done recently by the team, specifically Eli, Christian Stokes, and Stephen Tadas, to collect data, pre-process data, and reconstruct the data better, have addressed that and really proved the dynamic range of the system. So it's not just the sensitivity, it's also the dynamic range across the regions of interest within the reconstructed test subject. So I'm very excited about where this fits. And I'm going to go to kind of a summary slide here. It's very wordy, I apologize, but I think it's worth having a discussion of where CFT adds value in the and why frankly I'm so excited about it. And I have the general obvious bias of working on it for a long time and also coming to it from the perspective as a founder and director of the company. But I really would say now working at Ratio Therapeutics, which is a translational a targeted radiotherapy company, it is so useful. I think we're excited to use CFT to answer questions that can be harder to answer with radioactivity. So as a first bullet, cost and ease of use, CFT is just simply much cheaper and easier than radioisotope research, and it is dramatically superior to in vivo fluorescence and bioluminescence. Now, people have infrastructure, but it's just a lot easier to work with the fluorophore. In terms of imaging, sensitivity and resolution, and I'll address throughput, CFT produces better images in basically every metric. You saw that we could make very exciting C14 images, but that is not, it's a non-trivial task. And that's a caveat I'd say here, that you can do almost anything in nuclear, but it just might not be easy. And you can do a lot of things with a lot of technologies in spatial transcriptomics, MALDI, DESI, LASI. This is just easier, I would say, and it can be a real feed. I'll come to that in a moment. A real feed to other modalities. Throughput, I just want to add, quantification of CFT is excellent, and the team is working hard to improve it. The relative quantification, I think we certainly struggled with it in terms of settings, but I think the system now and if your protocol is paying attention to your relative and quantification is phenomenal. We're working hard to characterize the absolute quantification, but it will never compare to radionuclides. I mean, radionuclides are the essence of quantification and always will be. Throughput's not so obvious, as we discussed. There's clearly the throughput of bioluminescence and in vivo fluorescence in a benchtop scanner. People could do 20 mice in 10 minutes, but it's about information content. In terms of information content, I think the throughput here is phenomenal, very analogous to microspect and micropet throughput, if not superior. It all depends on the task and what you're setting up. In terms of chemistry, and I think we all know we're as good as our chemistry, there's a lot of trade-offs, but in general, fluorescence is just a lot easier to work with. And there's so many other exciting applications of fluorescence, with a major caveat that and you don't have to deal with half-life and decaying things and shipping. It's just, life gets so much easier. But all of those are surmountable, but all of them are a pain. In terms of a caveat, fluorophores are not 1.5 kilodalton. When you go below that, which is a real body of molecules being studied, you're not guaranteed that you can support a fluorophore or that you'll have anything fluoresce in that at 500 daltons. It is, on the other hand, a lot easier to use for. We could also say, I just think the labelings I've experienced when people have more success with fluorescent labelings of proteins, but there's always a way in nuclear imaging, but it can be, it's often a very heavy lift. And even if you can't put you can go to ex vivo nuclear too, that you can always synthesize a compound with C14 or tritium, or not always, but often. But that is also expensive and time consuming. So I just think the chemistry it just opens up a lot of doors for research. On the flip side, the translation to clinic, let's be clear, radionuclides are perfectly suited to translate to the clinic, and there's really no real comparison here. There's a movement in fluorescent guided surgery, and we have had a lot of conversations about it, injecting a fluorophore in that context, and then resecting a tumor and then studying it. But that's let's be open here. Radionuclides are the best translational molecular imaging modality, and always will be. In terms of translation of other assays, CFT is, again, so much easier to do downstream work, to move to microscopy. Once you have radioactive tissue, it just makes life very difficult on a lot of levels. You can decay and move on, but the fact that you can have the fluorophore in the test subject and then study this in other modalities is great. I will say too, moving from the Xerra system to a microscope, you can, those trained in the art can get tissue and study microscope. It's far better, in my observation, to just have a separate cohort or group that you're going to study under the microscope, as I alluded to earlier. And the notion of studying transgenics and pharmacoexpression, I mean, I think we shocked a lot of people looking at what the distribution of fluorophores in those two arenas, where people had a perception of what was happening, and then they really saw what was happening, and it wasn't what they thought. I think we made some people happy, and we've made some people upset when we've looked at GFP expression and other expressors in the modality. So I'll conclude with, before we go to questions, with just a huge, I won't be able to capture everyone here, 50, 70, a lot of people have worked on this, but I see some of them on this call, and there's been a huge, the great team at MIT, which has put forward just a solid system. There's a lot of growing pains getting this modality to be a product, but they deserve heroic cheers. And tons of people, some from the in vitro realm, and I want to thank all the people in the Biogen collaboration, as I really dove deep into that, as well as many other collaborators who were part of those studies shown and those beyond, and the development of the modality. So that's it. I'm happy to take any questions, and I hope that was informative.
Thank you, Jack. That was a great overview. We have quite a few questions that are coming through the chat, so I'll ask those to you. And thanks for the direct comparison to PET. I know a lot of the participants are in the call are directly work in pharma, and I think that really gave them a really good insight in where CFT fits and how to directly compare it to those standards that are widely used today. So I'll just go with a couple of questions. So we have about 10 minutes for questions. Feel free to send those in through the chat line. And I'll start with the first question, which is, how many fluorophores can you image simultaneously? What's been your experience?
So I know the system, and if someone pregnant, you could do up to 6, but typically 2 or 3 would be the rational. Very analogous to multiplexing IHC. You can do 6 there, or okay, some 5, but that's the capability in the standard.
Great. And then a question that I haven't seen before, which is there a possibility of imaging Cherenkov luminescence during section?
Yeah, that's a good question. I'm not sure. I just don't know that when you think about Cherenkov luminescence, and I'd love to try it. If someone knows from the in vitro or mid team, let me know. My prediction is no. I don't know. It would be great to try. It's a very good optical imaging system. But I'm not sure that it can get down to that low number of photon count. But it could. It would be great to try. And certainly you could have more radioactivity there.
Next question.
My apologies. I've thought about that and wanted to do it, and we've never done it.
Are there any particular types of drug compounds?
I asked by Matt Silva. He's the grandfather of Cherenkov luminescence imaging, as he discovered and developed, as he developed it at Millennium.
Are there any particular types of drug compounds that you think CFT is most suitable for?
Yeah, I mean, it really is a peptide to nanoparticle powerhouse. There's really, it just is great for any compound in that regime. Our compounds at ratio are around 1.5 kilodaltons, and that can support a fluorophore, but you want to be careful. Again, small molecules do not always work in this context, but any biologic is great.
And then one question is, we're currently using PET as our main imaging modality for drug discovery today. how would you go internally and validate that CFT can be an imaging modality to consider instead or in addition to PET? And for what kind of experiments?
Yeah, and I of course love micropet and microspec, but I actually just foresee using CFT more and more and replacing it. The first would be to just have study the biodistribution of a compound with a radionuclide and then study it with a fluorophore. That's the ultimate validation, and just do a comparator of the analyses. The image quality will obviously be much better in CFT, but that's the validation. We know that works, but if I were you, I would want to do that experiment to feel comfortable. That's with all the caveats that you have to make sure you're confident in your labeling too. That's a precursor. That's no pun intended. That's a given to validate that. But sorry, go ahead, Catherine.
Yeah, no problem. How do you go about quantifying biodistribution with the system?
Yeah. So as I alluded to, the relative quantification and trending to percent ID, trending to a percent ID per gram, it's I think we'll get there. Right now, the relative quantification is very good. So comparing an organ to which is actually the standard for most nuclear imaging quantification tasks as well, is comparing to a reference region and comparing to the whole animal. The biodistribution sentiment here is to go by region and look at the some signal and concentration of the signal in that region of interest. And it just doesn't have the same. Nuclear is inherently more quantitative. We hope to get to that kind of nanomolar concentration per voxel. It seems very, very likely, but that's where it stands now. It's very much a relative quantification. Yeah. Certainly relative to what's in the whole animal is very easy to do.
Right. And then maybe a little bit repetitive to what you mentioned before, but this question came through. Are there specific fluorophores that are more recommended?
Well, that's a great question. And, I don't know if I'm the best person to be in that debate. Clearly, as I alluded to, the extinction coefficient times the quantum yield is the figure of merit in terms of brightness or subsequent photon production. There are some other trade-offs. You have to look at, when you're in the near IR, you're not going to have a lot of a background. If you're in the green, you're going to have a lot of background. So you just have to ask yourself the task for that experiment and what is the right fluorophore selection. And then I will let the fluorophore gurus debate the properties of each one. So, but those are the guiding principles.
Okay. Well, I think we're coming to the end of our time today. I believe I've answered, there's quite a few questions coming through, but I think I answered all of them. If I missed your question, please, I'll take another look and email you directly if there's anything that we forgot. I also want to mention that please make sure that you've registered on the link I provided at the beginning in order to make sure that you receive a copy of this recording. And it's great to have seen so many people dial in today. And thanks again, Jack, for that great overview and presentation. And we look forward to hearing more about the advancements you're making in the future.
Okay, does Daniel Bradley have a question? I see his hand raised. Oh, his hand went away.
Oh, let me check.
Nice hand raising, though. I'm proud. I saw that. I've been a Zoom disaster today, so it was good. But listen, thanks. It was great to see everyone's names. It would have been more fun to see you in person. A lot of great colleagues on the call, and I hope that in society we're able to get together in person sooner than later.
Great. Thank you, Jack. Have a good rest of the day. Bye-bye.
Have a great day.